the assembled viruses, they leave their host cell. They leave the host cell in one of two ways, okay? Naked viruses leave the host cell through cell lysis, okay? So that means that naked viruses, they kill the host cell immediately. They right away kill the host cell right away. Enveloped viruses are released from the host cell by budding or exocytosis, okay? Meaning that as they are leaving the host cell, they take with them a part of the plasma membrane, which is why they get enveloped, okay? I want to make a point here that enveloped viruses, this is a question that students always get wrong. Enveloped viruses, an enveloped virus gets its envelope from the host cell during this stage, the release stage, okay? And enveloped viruses, they get that envelope from the release stage from the host cell. This envelope is a lipid bilayer, and they can get this from the host cell plasma membrane, from the host cell nucleus, because remember that our nucleus is also a lipid surrounded by a lipid bilayer. Okay. Okay. So this is a summary of virus life cycle. Okay. I want you guys to know the following. Okay. So again, the attachment phase, viruses are going to attach to a host cell receptor through viral attachment proteins located either on the viral capsid or the envelope. I want you guys to know these examples, okay? The hemagglutinin of influenza virus. So the influenza virus has hemagglutinin receptor, okay? The HIV virus has the GP120 receptor, okay? These bind specifically to host cell receptors, okay? What are they? Sialic acid, this is the receptor that the influenza virus must bind to. And then the CD4 receptor located on our white blood cells. Okay, you have to please know these. Okay. Okay, the other thing is that nucleic acid replication. As we said, DNA viruses replicate their genome in the nucleus, except pox virus. All DNA viruses replicate their genome in the nucleus except pox virus. That means that pox virus replicates its genome in the cytoplasm. It's the only DNA virus that's an exception to the rule. All RNA viruses except influenza virus and retrovirus replicate their genome in their in the host cell cytoplasm, which means influenza virus and retroviruses are the only RNA viruses that replicate in the host cell cytoplasm. Okay? Very good. And then the assembly and the release, which we already went over. Okay. This here we're going to go over steps in viral replication process. Steps in viral replication process. Okay. So,
Dr. Simmons, you said that RNA viruses, except influenza virus and retrovirus, replicate in the cytoplasm. But then you said again that influenza virus and retrovirus replicate in the cytoplasm.
whoa. Say that again. You're
When you said that all RNA viruses, except the flu virus and retrovirus, replicate in cytoplasm, you just said, like, you basically repeated it, but you said that the influenza and retrovirus do replicate in the cytoplasm instead of,
oh
like,
no. Then I meant to say that they replicate in the nucleus. Yeah.
right. Okay.
Did I say that? I didn't realize that. Good catch,
Yeah.
though, Fadi.
Okay.
Good catch. Good job. I should give you bonus points for that. That's very good. I didn't catch myself. Very good. Good catch. Um, very good. Okay. Um, so again, we were talking about how viruses can invade and enter a host cell. Okay. Obviously, in order for viruses to invade a host cell, they need a route of entry. Okay, route of entry. For example, many viruses enter the host cell through the respiratory tract. For example, the coronavirus SARS-2 virus is transmitted through, the respiratory route. Okay. Other common portals of entry are the GI and the urogenital route, mucous membranes, or sometimes even breaks within the skin secondary to trauma. Okay. Once a virus enters through any of these routes, it undergoes replication, and the replication occurs at the site of entry. Okay, it occurs in the site at the site of entry. Then it can disseminate or spread from the primary location through which it entered, and usually it disseminates through the circulatory system. Okay, so viremia basically means viremia means that viruses are present in the blood. Some viruses also have the ability to travel to the lymphatic and nervous system as well. Secondary replication. So when upon dissemination, secondary replication can also occur as well, which means that secondary viremia can also occur as well. Target organs. So this is referring to basically which tissue tropism or which organ does a specific virus have affinity for. Okay. The virus is shed into the environment. Okay. For example, through the respiratory route, by coughing, by sneezing, it gets shed into the environment. Okay. Usually with viruses, they usually die when they are outside, when we sneeze them. Apparently, with the SARS-2 virus, it has the ability to live on inanimate objects like on pieces of wood or on metal for a number of hours. Okay. And then other people come and touch that area, they can possibly get infected with it as well. Okay. So now, um, so viral damage to the host cell. Viral damage to the host cell. So